welcome to all of you to join this session. I am very happy and I have great pleasure to introduce today's second session resource person, Sowdhamini. Now before that, I want just to introduce one thing regarding this department. Bioinformatics department, they have this proper planning and their main objectives. They have produced many meritorious and outstanding students working in all over the country. And I think tomorrow and after tomorrow, this department become number one. in that way, this department travels. Okay. So with this background, I just take this opportunity to introduce our resource person, Sowdhamini. She is a very distinguished and well-experienced professor from a prestigious institute, the National Center for Biological Science, Bangalore. She leads a group whose broad interests are in computational approaches to study proteins. In particular, her group is interested in protein domains, folds, protein-protein interactions, protein-ligand interactions, virtual screening, genome-wide studies, and in sequence-based detection of distant relationships. Of course, she has good academic and research careers also. Sowdhamini was trained in basic chemistry and she acquired her master's in Indian Institute of Technology, Chennai. And she pursued her PhD at the Indian Institute of Science, Bangalore. Subsequent to her postdoctoral tenure at Professor Sir Tom Blundell at Birkbeck College, London, and later at Biochemistry Department at the University of Cambridge, she joined NCBS in 1998 and has been pursuing her interest in genomics and protein structural analysis. Sowdhamini is internationally known for her inputs in the classification of proteins, fold similarities in protein structural domains, and in the application of protein structural motifs to recognize relationships at the superfamily level. She has developed methods in the laboratory for recognition of distant members of protein family and small molecules that could abrogate protein-protein interactions. She has now 2,200 publications in international journals and has guided 23 PhD students so far. So with this, I welcome Dr. Sowdhamini to take the class, and I request each one of you to listen this session and interact, so that this session also make it in grand success. And I think one of the topic is she is interested in protein folding. Just I want to raise my question. One of the main questions always raises even LKG students. Why we need water? Because this topic is interested. The folding structures of this protein is very, very, very important. So just I raise my question: why we need water? Even any living species need water. Anybody give answer for this? Why we need water? വെള്ളം എന്തിനാണെന്നാണ് ചോദിക്കുന്നത് മലയാളത്തിൽ പറഞ്ഞത്. Even LKG students, they raise questions because most of the topics relate to this. Anybody raise? Because we feel that thirsty and we take water. Okay, why we need water? അമ്മേ വെള്ളം എന്തിനാണ് ഞാൻ കുടിക്കേണ്ടത്? എന്തിനാണ് വെള്ളം എനിക്ക് തരുന്ന അമ്മ? ചോദിക്കുമ്പോഴേക്കും നമ്മൾ എന്ത് ആൻസർ പറയുന്നതാണ് കുട്ടിയുടെ അടുത്ത്. Why we need water? It's a very good question, and this. Now I conclude like this. Most of the Nobel laureates in chemistry concluded like this, because this department is bioinformatics, information related. You see, we can think, we can live even 2 days or even 1 week or even 1 month without water and food. But in future, we cannot live even a single minute without blank. You fill the blank without. And so we need information, okay? Information is very, very important. Without information, I think So even, this water, what is the function of water is nothing but to give a folded structures of proteins, because we have many thousands of proteins, each proteins having its own role, and this role coming from folded structures. So protein may be dancing, jumping, walking, and all these things. So folded structures coming through hydrogen bonding with water. So this is what we need, water. Okay. So I just stop and I welcome to this madam to introduce this.
Thanks for the very nice introduction, and thanks to the Department of Bioinformatics and the University of Kerala for inviting me to share some of the research interests. We've had an excellent time and been hearing and seeing that lot of talent hidden with the students here and the faculty as well. But today what I thought I will share some of recent interests on plant genomics. But before that, this thought-provoking question about why water. It's very true, because water is required to keep the proteins in control, that globular shape. But also, most of the human body is made up of water, and that pressure is required to drive the chemicals in the right place. So there must be many more reasons. So I think, however, I'll come back to my original planned topic of talking about plant genomics today. What I intend to do is to share some of our experience dealing with the whole genome sequencing of a medicinally important plant, a herb called tulsi. In the European term, there is an equivalent plant herb which is called as basil. And why plants are interesting? We got interested about 2 years ago looking at plants, and especially the genes and the proteins behind them, because of our own thought-provoking appreciation that plants are there to give us many things. They give us shade, they give us food, and they give us seeds for their own reproduction, a long life, and so that it can go on for many generations. But also it gives secondary metabolites. Secondary metabolites by definition are not entirely required for the functioning or the development of the plant itself, but it might be used for defense. But certainly humans exploit them greatly. Time and time again, over 3,000 years in the stream of Ayurvedic science, especially in India, people have been using plants with their knowledge about which plant is useful to heal what sort of diseases. And this is known time and time again, and it's never questioned. In fact, we are fortunate if the knowledge is passed on from generation to generation. But as scientists, we often have this question as to what is the chemical behind this? What are the genes that produce these chemicals? So with that sort of a question in mind, I started off looking at plants with to see what sort of metabolites are produced. And in this slide shows a picture of the different unique metabolites that are produced by different plants. Tomato has a particular chemical, and then maize has a separate chemical, and you will be amazed to see that often these are endemic to the plant, and one chemical which is produced by one is not exactly produced in the same amount by another plant. And also if you look at these small molecules, you can see several chiral centers. They're amazingly complex, but the plant is able to produce them without error and in large amounts. That's how humans are able to exploit them. So we could look into whole genomes of plants and see if we can identify what is it that render this sort of a uniqueness or capability to produce a metabolite in large amounts by a particular plant, and whether there is any overlap and what is different about these of the plant enzymes that make some of them to produce a chemical in large amounts, whereas not the others. Of course, these questions remain, but it's our beginning and attempt to go and look seriously in plant genomes for that purpose. And we also wanted to include evolutionary angle and see if there are amino acid changes near the active site of such enzymes, which could explain the tremendous efficiency in comparison to other plants, and so on. We chose tulsi mainly because it's the queen of herbs. It has a multitude of chemicals that are produced in large amounts, and that has been exploited by humans over many generations. And as the name suggests, it is being used for a variety of curing purposes, whether it is tuberculosis or fever or diabetes or cancer or infection of multiple sorts. And indeed, when we look into this particular species, we find that there are subtypes, or there in fact are many subtypes of Tulsi, and the most common in India is the Ocimum taniflorum, and here itself we have two subtypes, the Rama and the Krishna types, and the Krishna one has a dark purple coloration in its leaves, whereas the Rama one has a light green color, and both of them have been implicated in Ayurveda, and in fact over thousands of years these leaves are kept in holy water and distributed to the society whenever people come to Hindu temples, suggesting that this is a preventive mechanism to avoid infectious and contagious diseases, and this is practiced even today. And there are other subtypes like Vana Tulasi, Karpura Tulasi and so on, and from the point of view of genomics, what is interesting, it is diploid, and its genome size is estimated to be about 612 million base pairs. And since this is the case, we went and looked in the literature to see, Ayurveda science aside, can we look from an enzymatic biochemistry perspective and identify which are all the metabolites that are responsible for this high degree cure and medicinal value. And we kind of researched, looked around 300 different papers, and there are books of Tulasi and there are reviews of Tulasi, so it was not very hard. And there are already many papers that suggest clearly what are the metabolites that are responsible for the cure of a particular disease, and there were at least 43 of them. And we decided to look into a database called PlantSeq. It is a little bit like CADE database. It will record the pathway of the synthesis of a particular biomolecule. It could be a small molecule, it could be a secondary metabolite and so on. And we were able to trace the pathway for the 43 different secondary metabolites, but for only 14 of them we could clearly identify the genes involved in the synthesis of the metabolites that we know are present in Tulasi in large amounts. So this aside, then the very important question came as to, can we identify these genes knowing that they reside in Tulasi plant? And we had chosen about 14 of them for which full pathway information was available, but that's where our interest in superfamilies could help. And we also know from our own experiments that the metabolites indeed are in large amounts. For instance, urusolic acid and eugenol are implicated in anti-cancer and anti-infection respectively, and you can see that if we do a mass spectrometry and a metabolite confirmation, indeed the Rama subtype has the highest amount of eugenol in the leaf, and you can see it in the other subtypes, they are not that high. So this clearly explains why in a Hindu temple Rama leaves were used in the holy water, but then this happens also for the urusolic acid. You can see really high amounts of urusolic acid in the Rama Tulasi leaves. In comparison to any of the others, this has the highest amounts. So the chemicals are there in high quantities, but can we use PlantSeq database to know which are the genes involved? And for instance, for urusolic acid, it's known starting from alpha amyrin, which is a small metabolite. 3 different enzymes are used to give rise to this very complex molecule called urusolic acid, and it is involved in anti-cancer. Whereas for eugenol also there are a few enzymes involved. But my interest on superfamilies really started with my postdoctoral tenure in Professor Blundell's group, where we began looking at protein domains in different families, and were able to appreciate that indeed irrespective of the sequence diversity and irrespective of the fact that certain sequences that may be only 10% identical, they can still retain similar structures, 3-dimensional structures, and sometimes bear similar overall function. So this is a term superfamily. So we were in the lab very excited to see if we can look for these genes in the Tulasi genome. And then the very obvious next question is, do we have the Tulasi genome with us? And we did not, and that did not stop us from looking for it, because we were so excited about this plant. And I asked my lab people, how many of them would be interested in going through the exercise of the next generation sequencing, getting the DNA assembled and the genes annotated and so on. And to my surprise, everybody in the lab raised their hands. And from then on, we used to hide in some seminar labs every week, and we used to discuss about this particular project, which was tremendously exciting. And we found ourselves meeting every day towards the time we were looking for the enzymes of interest. And this is just the bioinformatics group. We also collaborated with CCAMP and INSTEM. These are neighboring institutions where the DNA was sequenced. And you can see there are 3 people from Kerala here, and Agnel is right here with us, and Sony is also right here with us. And of course, I am somewhere here. So this is the kind of little history about the people involved, because everyone wanted to learn about the next generation sequencing techniques, the assembly, the annotation. So we started from scratch, and that's what new things we were going to do, because we were used to searching for distantly related members, the superfamily members, but this sort of chemistry and genomics bioinformatics was new for us. We wanted to purify the DNA of Tulsi leaves and then also include the transcriptome data from many different subtypes, both the Rama and Krishna subtypes, and then see if we can identify the metabolome of Tulsi, as it were. It's not a complete, because there are at least 43 different metabolites, and we could place hand on only 14 of them, but at least can we identify the enzymes that are present in the pathway of the synthesis of such small molecules. And in doing that, we had to also include evolutionary data, because we wanted to compare with the genome of monkey flower, which is the closest whose whole genome data were available. And also we wanted to compare with tomato, potato, and the very well characterized genome, of course, which is the Arabidopsis. So I'll just take you through 4 or 5 slides, which gives the methodology of how assembly and annotation is taken through, typically for a plant genome for which we do not have a reference. So essentially we have to assemble the genome bits by means of de novo techniques, by which we do not have any reference closely related genome you can use to stitch the raw reads together. But then we had, to our advantage, 2 libraries that were used by the CCAM colleagues who are our collaborators, and they had used both paired end libraries and the made pair libraries, and each one of the reads were fragments of the DNA which were about 100 base pairs long. Of course, before we start to assemble the data, the very first step that people do is to clean the data, and we had 90 gigabases of paired end data and 26 gigabases of the made pair data passed on to us, and these were the raw reads. And then we proceeded to apply some filters using FastQC algorithm, by which we wanted to trim off the unreliable data. Only the statistically significant data reads were kept, part of the reads were kept and used for the assembly process, and this was done using FastQC. And then we were able to use variable nucleotide windows, which are typically called as schemers, by which you have to start off with as small as 2 or 3 nucleotides, and we started with 13 nucleotides, and we keep changing the frame so that we see what sort of nucleotide stretch gives rise to best assembly. And the raw reads are all stitched in the process, and the validation of which schemer is the best is measured by means of a couple of parameters. One of the most important parameters is called N50. N50 is analogous to the IC50 values, which we are worried when we look at biochemical activity of enzymes, right? Because how much concentration of the inhibitor is required, wherein you have 50% of the inhibition. So also here we would like to know what are the number of nucleotides in the 50% average size of the scaffolds which we have just been able to assemble. And scaffolds and contegs are nothing but the higher order stitch regions of the raw reads, and larger this number, the better is the assembly. And we find that the best assembly was produced using a 33 kmer window frame, and then we did the same for the made pair DNA as well. made per library as well, and then we were able to arrive at a optimal chemo value, which gives rise to about an assembled genome, which comprises of 363 million base space. And this corresponds to about 53% coverage, but together with the transcriptome data, we were able to get 61% coverage. And if we do validation, which is all the raw reads are mapped back on the assembled genome, we get about 75% of the reads or coverage with respect to the raw reads themselves. And then we did something which is to use the singletons and the mate pairs together, all together for the assembly process. And in this case, we use a program called Sowdhamini 2. And this is a very useful program if you do not have a reference genome, as I said before. And subsequently, we had to filter out repeats. And this is a feature of many plant genomes. Many plant genomes have a large number of repeat regions that have to be masked. And this was done using a program called Repeat Masker, which iteratively learns new repeats that have been just identified in the plant of interest, and it removes them from the annotation process. So after assembly, we remove repeats, and the next process is to actually look for the genes of interest. You might think that gene prediction programs are nearly ideal, but as we come across this procedure, we realized how complex the gene prediction algorithms and the gene models themselves are, because we find that for eukaryotic organisms and especially for plant genomes, many features are not as ideal as it seems. You do not have the right patterns to unambiguously recognize gene models. But then we used a program called Augustus, and then we used Arabidopsis genes to train the algorithm. And using this approach and combined with more than 10,000 scaffolds of the assembled genome, we were able to use transcriptome data as evidence further to recognize genes. And this turned out to be a very extremely debating time, because almost every gene we used to sit and debate as to whether this is the right gene model or not. And even to arrive at these algorithms and the training genome was not straightforward. We had to do trial and errors, and this is a final flowchart that seems quite appropriate for this particular plant genome at least. And when we do, we were left with 36,768 genes, and these numbers more or less match with what we have for Arabidopsis. So the first news that we realize is that it's not as though Tulasi genome has a large number of genes, something that makes it special and renders its medicinal value. And then in terms of the RNA transcripts, we had been able to assemble them separately, and they also gave rise to quite a few transcripts, and about 40% of the gene models had transcript evidence as well. So this is the methodology part. And from here, I would quickly go back to our interest on the metabolites and the metabolite synthesizing pathways. And we also had to ensure that the genome predicted genes were as good as any other plant genome. And for this, we consulted a database of essential genes, and we found for nearly 93% of the essential genes we were able to find in the assembled genome, and the genes that have been predicted by our computational approaches. And subsequently, we asked the obvious, which is to know how many PFAM domain families are there in the 38,000 genes that have just been identified. Are we getting similar numbers? And here again, we found that there are a large number of protein kinases and the LRR domains. And in fact, Tulasi genome and many of the plant genomes indeed have large number of the cytochrome P450. And it turns out cytochrome P450s and the terpene synthesis are large superfamilies that are being used over and over again in a variety of contexts by plant genomes. Not only Tulasi, but many plants that are medicinally important use these two superfamilies a great deal. And we also find them occurring to a large number in the assembled genome. And then we also found, when we compare the composition of the genes with respect to 23 other plant genomes, we find the composition of the orthologs. And these bars indicate how many orthologous relationships could be observed in how many other plants. And higher this blue bar means These are unique genes because the orthologs could be observed in only 2 other genomes out of the 23. And if it is more of the orange, it means such genes, and the length of the bar indicates the percentage of the genes in the genome, and such genes are observed in all the other 22 plant genomes. So we can see that the Tulsi genome is somewhere here, and the pattern of the orthologous composition is rather similar to that of the monkey flower, the tomato and the potato, suggesting that again not only the number of the genes are similar to Arabidopsis, not only the domain composition is like other plant genomes, even the extent of the homologous family relationships the genes share with their neighboring plants is rather similar to that of the other non-medicinal neighbors. Okay, so then we chose to concentrate on the genes that we suspect are involved in the synthesis, and we call them as metabolite genes. From here onwards, I refer to them as metabolite genes, and we came across 438 examples of genes in the genome, in this particular genome, and then we asked the question how similar it is to the neighboring genome, say of tomato or grape or medicago, which is the monkey flower. Sorry, medicago is another model organism, and Arabidopsis thaliana. And the circle inside and the colors indicate the chromosome numbers for the other plant genomes, and the circular lines indicate that they all have a ortholog in the other plant genome. And we find that all the 438 metabolite genomes and the way they get organized, and if we project their dissimilarity as a phylogeny, is no different from the entire phylogeny of the different plants themselves. And this suggests that the medicinally relevant genes, the genes that are responsible for the secondary metabolite synthesis, and your phylogenetic variation is not different from the entire variation of the whole genome itself. So what is happening? What is different about Tulsi? So either the number of the genes must be more, or the expression of the enzymes that are required for the synthesis must be tremendously high, or the enzymes must be tremendously efficient, isn't it? Because the genome and the number of genes are more or less the same, the number of domains we see and the domain distribution are the same. Even if we concentrate on the metabolite genes, their phylogenetic distribution with the neighboring genes are more or less the same. So then it could be any of the three at least. So then we sought out to use the transcriptome data and then ask the question, can we use the transcript information and see how much of these genes are being expressed? Are they being expressed in high amounts? And to address this, we used the transcriptome data, and sure enough, we found that for various 14 different metabolites, for example, the luteolin pathway, we have flavone synthase 1, naringenin oxidoreductase, flavone monooxygenase, naringenin hydroxylase, and NADPH oxygen oxidoreductase. These are the 5 enzymes that are consecutively involved in the synthesis of this very important molecule called luteolin, which is present in Tulsi in large amounts. And we find that in fact when we go and look for the genes using our typical sequence search algorithm approach, we find that there are 7 hits for the first enzyme, 9 copies for the second enzyme, 5 copies for the third enzyme, 26 copies for the fourth enzyme, and 7 copies for the fifth enzyme. Not only that, the expression levels as evidence from the transcript data and when normalized for all of the genes and expressed as RPKM values clearly suggests that they are available and expressed in large amounts. High values, even anything more than 4 RPKM values means it's significant, and we can see that the average is like 34, 61, 5, 104 and 59 and so on. So this is one example, but this sort of an exercise was taken up for each of the enzymes that we think are involved in the synthesis of the 14 metabolites. And this is the second example I have here. This is chopped out for eugenol, methylchalcanol and linalool. And again you can see very high values of the genes that we think are involved in the synthesis of such metabolites. And the third example is that of Taxol. Taxol is an anti-cancer compound. In fact, Years ago, it has been already recognized as important for anti-cancer, so much that there is an industry that synthesizes it. We are not depending on plants like tulsi anymore, but there are chemical industries that would go about synthesizing Taxol. And here again, we find that a host of enzymes are involved in the pathway, and their presence are high, not only in the number of copies, but also in terms of the RPKM values. That shows that a simple plant, tulsi, how it's able to synthesize secondary metabolites that have been exploited over years for the benefit of humans and to cure human diseases. This sort of an understanding is just a first step. As you can see, this can give rise to huge amount of commercial utilization, which we as fundamental researchers are not yet prepared. But given that these genes are present in multiple copies, we can in fact go after them, clone them, overexpress them, and in fact the big vision is to be able to engineer it in easy to produce and maintain model organisms such as yeast or E. coli or even Arabidopsis. Our little baby step towards this is to find out if not only their expressions are high as evidenced by transcript data, are they also in high numbers using RT-PCR experiments. And in this we selected a few of them that are involved in synthesis of uracolic acid and eugenol especially, and a few of them like taxadiene synthase, and compared to the control and few other genes. The next slide shows that indeed certain enzymes like the last enzyme of the uracolic acid pathway is observed in very high numbers observed using RT-PCR experiments in Rama leaves, especially in mature Rama leaves, compared to the young leaves or the stem or the other subtypes such as Vana tulasi or Karpura tulasi. This kind of provides scientific evidence and conviction for scientists like us to believe not only what the elders have said and practice it blindfolded manner, but to realize that indeed the metabolites are there in high amounts, indeed the genes are observed in multiple copies and in rather high amounts. So I was very happy when this was this had reached the stage. For no particular reason we were in a rush, because we were so excited, we were meeting almost every day. Little did I realize that the plant genomics or any genomics projects are as competitive as protein crystallography projects, because as we were working on it, the same city, Bangalore, Genotypic was also solving Tulsi genome, which we didn't realize, we didn't know about it, and it was in collaboration with the Center for Medicinal and Aromatic Plants now, and that's no surprise. But to our advantage, we had deposited the data earlier than them. We published a little late than them, but doesn't matter because we can work together now. At least that's how our lab views it. But then we put up this note, we got our paper published in BMC Plant Biology, and then decided to put it up on the news, and to our surprise we had a very good response from the press. This got published in Hindu, not only in Hindu, but then it came in many other, 7 other newspapers, NDTV, Times of India, and several news channels kept calling me and said, can we interview your group? And we did that, and I ended up speaking in Kannada to one channel, Hindi to one channel, and then I spoke in Tamil to a Tamil channel, and I was almost going to talk in Kannada, which I'm not very good at, but luckily they told you can speak in English. So I had been involved in such activities which we never anticipated, but it was a tremendously exciting period, and I know we have just taken a first step towards it, but there are more plant genomes which I think I cannot talk about given this experience, but we hope to do more. I would like to thank all the people involved and take any questions which you might have. Thank you.
That was fantastic, Sowdhamini. I was just wondering how close the genes that you'd actually classified under the same name were. Is it possible that the plant has duplicated them to get slightly different functions in related synthesis?
Yes.
Or are they all on the same synthetic pathway?
Yeah. In fact, we are now cleaning up the data. Although we say there are 26 copies of a particular enzyme, you are very right. that in fact they may belong to the superfamily and they are all responding because of our sensitive sequence searches. We are now cleaning up the data in the sense looking back at the genes and putting them in the phylogeny of that superfamily and ensuring it sits in the right cluster and being able to recognize the other enzymes which are doing slightly other functions which might have been picked up by chance.
In your initial data, you said that Ram Tulsi had the higher concentration of this. Why did you go for Krishna Tulsi? Is there some reason why you have gone for it?
Well, that was the way things started, because it's a collaboration, right? Because the CCAM people had already chosen Krishna Tulsi for the DNA purification and the assembly. The deep genome assembly was done only for the Krishna Tulsi. Now we know that the Rama Tulsi has more of the metabolites and more of the genes themselves. But at that time it was not very clear.
And second is, I'm a computational biologist, so I can understand what is the difficulty in how confident are you in this genome that you have got, because there will be so much of computational things that you will be.
Yes.
So how much confident are you, and how do you think you can improve on the genome that you have?
I'm glad you asked me this, because we have to validate in a variety of ways. One way of validation is the database of essential genes and the Sigma database, which shows that certain genes that are bound to be present in plant genomes, are we finding them? Are we getting good gene models of them? And we find that nearly 91% or 93% of such essential genes we are able to find. And we also make sure that some negative controls are there. Certain genes that we know are in the chloroplast, we don't find them. And we also ensure when we search for genes that we have an e-value cutoff, very stringent e-value cutoff, and a query coverage filter, so that we are not seeing spurious suboptimal alignments, but nearly the full length of the query is being aligned to the genes of interest. And we also do another level of validation, which is take all the raw reads that we got in the beginning, and if we were to map them back onto the assembled genomes, are we still left with a whole lot of raw reads which we were unable to use in the assembly? And we are finding that 75% of the raw reads are indeed getting aligned with the assembled genomes. So these are typically some of the ways that people use for validation, but there should be more.
Still, that we have achieved that value over 180, that RPKM value. Madam, what is that RPKM value, and how you standardize that as a?
This is just like how for microarray data we look at more than threefold or less than threefold as highly upregulated, highly downregulated genes. In the area of RNA sequencing, it is already established. By means, here we combine the reads of Krishna subtype and the Rama subtype. We deliberately combine the reads, we assemble them, and then we ask the question, in comparison to the entire set of genes that we have, do we see any of the genes that are getting upregulated? So this is the fraction of the genes that we see, million reads, that we see more in the certain genes compared to the entire assembled pool. So we have, let us say, the entire audience here, they are all mostly sitting and listening. And at some point, certain people kind of get up and ask a question. So also here we want to use the entire data as a way of normalization and see if certain raw reads that are always million reads are getting associated. If we have a value of RPKM4, it means 4 million reads got associated to the gene of interest, which is pretty significant. And we get hundreds and 300, they are very significant.
How will we fix that 4 or 5?
In comparison to the full data, when we combine them. Yeah.
Next anxiety regarding red color of that. I don't know whether we identified the red color of that particular.
We did, yeah. Actually, that's how the anthocyanin producing genes. I didn't allude to it, but we had this anthocyanin biosynthesis genes that was indeed highly upregulated in Krishna, but not the Rama subtype. So this was observed.
Just we want to confirm that red color is coming from some chromophore or something like that. So
yeah, the green bar is for anthocyanin, and you can see in the Krishna subtype it is tremendously high. It was used as a control to ensure that our reads are coming up right for the RT-PCR experiments. And also we had used some essential genes like alpha-tubulin, dehydrogenase and DNA-J, and they were also highly expressed in all of them. They were in fact used to normalize this.
I have a question. Thanks for a very exciting presentation. I have a question regarding the basic sequencing part. Since this is de novo, what is the ploidy value for Tulsi, and did it pose a challenge?
Yeah. Luckily for us, the Osimum teniflorum, Rama and Krishna subtype are diploid. But some of the others, like the Vana Tulsi, they are expected to be polyploid. Because they are more amenable to being wildly present. So they can get into polyploidy. But the Rama subtype itself was only a diploid, so it was little easy.
Just one question. Thank you very much for a very interesting talk. I was wondering, are you looking at the possibility, maybe in future, of reconstructing the metabolic pathways and regulatory circuits in the cell, or something along those lines?
Well, actually some companies have come and talked to us. So far we have shown interest and shared the data for the 14 metabolites which are already available in that paper. So there's nothing, no secret. But you can see the wide range of scope. There are in fact some anti-tuberculosis metabolites that we see, anti-inflammatory, anti-cancer. So the scope is very high. In fact, some of the companies have already approached us and we told them, just like how we tried RT-PCR for these genes, can they do for the others that are involved in a particular event like anti-tuberculosis or inflammatory. And in fact, one of the, what shall we say, the skin cream people, they are interested in the anti-inflammatory properties, and it's a very big company. And they are going to chase after all the genes that we think are involved in the synthesis. So the next step would be to see we can express it in a small and easy to grow model organism and grow them in large amounts. And if we are lucky, the chemical could also be produced in large amounts. So that would be a big dream.